Then we get past the idea of they're going to flip it once. And we get to the idea of they're going to flip it some number of times. And then usually the number of times is they'll come up with some ridiculously large number. You say, well, you know, is that really necessary? That usually engenders a discussion about resources and the value of time and using the resources wisely in experiments. So we get to the idea of you're going to flip it some number of times and you're going to count something, either heads or tails, it doesn't really matter. And then we get to the idea of how you're going to evaluate that. How are you going to decide whether to reject or fail to reject a null hypothesis? And then that develops into this idea that, well, we're going to make that decision on how unusual the results look relative to what they would look like if you knew the null hypothesis was true. So that's a key concept in making statistical inference. And then we start to discuss how do you know what the results would look like if the null hypothesis was true. And then I get them to develop the idea that, well, we could get a fair coin that we know is fair, and we could flip that a bunch of times and see what results we get from that, Jason. So there's lots of these programs on the web. Our classrooms are all wireless. I just pull up one of these programs on the web, and we go through a coin flipping simulation, and we look at, in this case, I've developed with them the idea, well, maybe our experiment will be we'll flip the coin 10 times and we'll count the number of heads. And then here's an example of repeating that same experiment 100 times with a coin that you know is fake. And we start talking, well, so what result here looks unusual? Well, 0 heads looks pretty unusual, or 10 heads looks pretty unusual. In fact, out of the 100 trials we did, we never saw that. And the nice thing, of course, about doing it on the computer is you can do it 5,000 times, you can do it 10,000 times. So here's a 5,000 run. And now you can see that, of course, it's unusual, but it's not impossible to get 0 heads out of 10 flips of a coin. It's just rare. And that then generates a discussion about, okay, well, so we recognize that it's not impossible. It would be quite rare. And if we had concluded that just because we saw 0 heads, we concluded that the coin is not fair, we recognize that we could be wrong. Because obviously, here's an example. You can do it. There's 5 times out of 5,000 it came up 0, 10 times in a row. So that brings us back to that idea of experimental error and errors in inference, and then we then have a discussion about just how often are we prepared to be wrong, in a sense. So that's that controlling of those errors. And I talked about the sort of standard in science being 5% at the time. And then we talk about that directional differences in your alternative hypotheses. So are we going to put all 5% down in one end or the other, or are we going to split it up? Basic statistics, but in a sense what we're really driving home here is the point of how would we know whether our results look unusual given that we know the null hypothesis is true. And that's important both because it makes a connection to statistics and it makes a connection to probability distribution functions and all the stuff that they're going to learn in their statistics course. But it's also important because it leads into a discussion of what is the control group in an experiment and what is the control group doing. Go ahead. So we then have a more general discussion about what is called statistic power, about these errors more because the errors are connected to each other and how they're connected to each other and what you can do to control them. We talk about the relationship between power and error and sample size, which is a critical component to experimental design, and this thing called effect size, which is the size, the difference in the effect that you're trying to observe. So if I have a control group and I want to be able to detect a deviation of the treatment, it's very, very small. So I have a very small effect size. In order to do that, I have to have a very large sample size. I develop those ideas for them. We then talk in more general terms about variation that gets in the way of testing hypotheses, so things that we're not really interested in, but it obscures our ability to detect the effect. So you use examples like if you wanted to test whether some diet pill actually leads to weight loss, well, different people have different fluctuations in their weight anyway, even in the absence of taking any sorts of pills, and so that variation gets in the way of a test of the efficacy of this pill. And we talk about how you might control that variation in an experimental design by grouping people. In that case, we would group people together, perhaps by their initial weight, or perhaps by their weekly fluctuations in weight in the absence of anything. There are a variety of ways to do it. And that's an area of experimental design called blocking, which is key to experimental design. And I'll show you an example of that gone badly later on in a second. And we talk about external validity versus experimental control. So this is the idea of doing an experiment that has some relationship to the outside world versus being able to control the variables involved. One example I use is back in the, help me out, Jonathan, here, maybe the 70s, when saccharin was on the market. It was an artificial sweetener. It was in